Hi everyone, for this video we will continue genetic inheritance for subtopic 4.2 deviations from the Mendelian inheritance and 4.3 genetic mapping. Deviation from the Mendelian inheritance. The example of cross that deviated from Mendelian inheritance is codominant alleles, incomplete dominant alleles, multiple alleles, linked genes, sex-linked genes and polygene. We will go through one by one after this. Codominant allele. Codominant allele is both alleles of a pair are fully expressed in a heterozygote. An example of codominance is the human M/N blood type. Within the M/N blood groups there are two antigens, M and N. Individual with the genotype L/M will have only the M antigen. Individual with the genotype L/N will have only the N antigen. While individual with heterozygous genotype L/M it will produce both antigen, which is M and N. Please watch the next video to understand more.
Genetics. This is another type of exception to Mendelism. The term codominance can be divided into two parts, co and dominance. We know what co stands for. It means mutual or together. Just like cooperation, which means operating or working together. The second part is dominance. And we know what dominance means. An allele that masks the effect of the other recessive allele is said to be the dominant one. This gives rise to a dominant phenotype. Now the question is, how do the two terms co and dominance come together to give us the effect? And what type of effect is exactly seen in the organisms? Codominance is the phenomenon where both the dominant alleles are equally strong and thus expressed in the offspring simultaneously. So the offspring will be heterozygous and will have both dominant genes in the genotype. As a result, the effect of both the alleles will be seen in the organism phenotypically. How do you think we got this chicken with checkered feathers? In such a case, we cross a black feathered chicken with a white feathered one. To understand this theoretically, let's say that we have the genes BB in capital for black colored feathers and WW in capital for white colored feathers. On crossing the two, we get a progeny with all heterozygous genotypes. Each offspring has one B and one W allele. That means each chicken has both the dominant alleles. And both the dominant alleles try to express themselves. As a result, we get both the characters expressed in all the offsprings phenotypically. Does all the chicken have checkered, or in other words, speckling feathers? Now this is like an exception to Mendelism, but not totally. Just like incomplete dominance, even codominance follows a pattern. The F2 generation is obtained in the typical genotypic ratio 1:2:1. And the offsprings in the F2 generation exhibit the same ratio phenotypically as well. We get one black, two speckled, and one white offspring in the F2 generation. This is the phenotype. And what about the genotype? We get one offspring with the genotype BB, two with BW, and the last with genotype WW. This is how codominance is seen in the chicken, which gives us a completely different phenotype. Codominance is observed in humans as well when it comes to blood grouping. Let's understand this in detail. How many blood groups do we have, by the way? Actually, there are many types, but we mostly take into consideration the ABO type. And this type divides as how many groups? There are basically 4 groups according to this system. These are A, B, O and AB group. Now let's say this is a single red blood cell. There are various components attached to its surface which are called antigens. These can be long chains of proteins or sugars. Now, in the case of the blood grouping system, the different groups are classified on the basis of these antigens. So, if the RBC has an A antigen, then the blood group is A. Similarly, presence of B antigen categorizes the blood group as B. In case of O, there are no antigens present at all. And now, can you guess which antigen will be present on the RBCs if the group is AB? That's right. The RBCs will have both the antigens A and B present on their surfaces. Now, getting back to our concept, how is codominance seen in these blood groups? In the case of human blood groups, the types A and B are both dominant, while O is the recessive type. Now, if the offspring has one allele for A blood group and the other for B blood group, then the phenotype obtained will be AB blood group. This is how codominance is found in humans in the case of blood groups. Now, apart from humans, there are several other animals that exhibit codominance. Cattle, for example, have roan coat color. This is obtained on crossing cattle having white coat color with cattle having red coat color. Many other animals have a skin coat with two colors or have patches on their skin coat, which are examples of codominance. But please note that not all animals with patches will always be examples of codominance.
Incomplete dominant allele. Incomplete dominant allele is a form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other allele. The example of incomplete dominance is snapdragons. Snapdragons show incomplete dominance in the inheritance of flower color. The heterozygous flower has a phenotype that is in between that of the two homozygotes. When the genotype of flower color for snapdragons is heterozygous, which is capital R and small r, it will show pink color flower, the intermediate phenotype for red flower and white flower. Please watch the next video to understand more.
Plasmic inheritance. Also, these plants are used by many geneticists to study incomplete dominance. So what does that mean? So far, we've studied only dominance of characters in the pea plant. What does this concept of incomplete dominance mean? Let's begin with understanding the name. Incomplete here means partial. Does it indicate that the dominance is partial? That's right. The dominant character expresses itself, but not completely. It means that the dominant alleles do not completely mask the effect of the recessive alleles. As a result, offsprings with intermediate characters are obtained. This is the reason why the phenomenon is also called partial dominance or semi-dominance. So let's get back to our 4 o'clock plants to understand this. In this case, if a cross of homozygous red and white flowered plants is carried out, then the resultant offsprings are pink in color. So the blend of red and white gives us a pink color. This is because the dominant allele for the trait red does not completely mask the effect of the recessive allele for the trait white. So when the alleles are expressed in the offsprings, they give an effect which is like a blend of both. This is the reason why we get pink flowers. The same feature is seen in a snapdragon flower. A red variety crossed with a white variety gives us pink flowers. However, this is not a complete exception to Mendelism. If the pink flowers are crossed, then the F2 generation gives us the typical 1:2:1 genotypic ratio. How does this happen? Let's assume RR in capital to be the genes for the red colored flower. Let WW in lowercase be the genes for the representing white flowers. On crossing the two, we get all the pink flowers with the genotype RW. Now till this step, we find the inheritance to be an exception to Mendelism. That's because Mendelian genetics did not talk about blending of characters. However, on crossing any two from the F1 generation, we obtain the genotypic ratio of 1 to 2 to 1 in the F2 generation. Even though the phenotypic ratio is not 3 to 1, the genotypic ratio obtained is the same as we had obtained earlier. So this pattern of inheritance seen in Snapdragon is an exception to Mendelism, but not entirely. Because the F2 generation still exhibits the genotypic ratio 1 to 2 to 1. And if you noticed well, this is the phenotypic ratio as well. Now this was about incomplete dominance in plants. Is this seen in animals as well? Oh yes, it's seen in human beings too. Out of the many characters that show this kind of pattern, hair type is the one which exhibits the pattern of the incomplete dominance. Curly and straight are the two types of hair traits which we know. Crossing these types gives us the third phenotype, which is wavy hair. This is nothing but an example of incomplete dominance. So let's try out the theoretical cross now. Let's assume CC in capital to be the genotype for curly hair. Similarly, let SS in lowercase be the genotype for straight hair. Here, the trait of curly hair type is dominant over the trait of straight hair, which is recessive. So can you solve this cross further to find the genotypic ratio in the F2 generation? This is what we get on solving this further. The F1 generation will all have wavy hair, that is, a blend of curly and straight hair. So all of the offsprings here will have CS as the genotype and wavy hair as the phenotype. And the F2 generation will be obtained in the ratio 1 to 2 to 1, both phenotypic as well as genotypic. This is how incomplete dominance is found not only in plants, but even in animals. So how does this help us? When it comes to plants, we obtain several beautiful phenotypes which can be used for many purposes. Animals exhibiting incomplete dominance are excellent models for genetic studies.
Multiple allele. Multiple allele is more than two alleles control for particular trait of a character. The example for multiple allele is ABO blood group. For ABO blood group, there are three types of alleles, which is IA, IB and I recessive. Please watch the next video to understand more.
There's a lot of phenotypes that are easy to tell. Your eye color, your hair texture, your height, whether you have a straight thumb or hitchhiker thumb. But one phenotype that you can't tell just by looking is your blood type. Your blood is really made up of many things: platelets, plasma, red blood cells. But you've probably heard before that when blood is donated, it's very important that it is matched correctly. And that's true because blood type phenotypes vary. It really boils down to the fact that red blood cells, they're not naked. They have proteins on their surface. And it turns out that your immune system is very protective, and if it gets blood that's donated that has different proteins that it's not used to, it will attack them. With blood type, you can have several different phenotypes: A, B, AB, or O. These letters stand for antigens that are found on the red blood cells. So type A blood, for example, has A antigens on the surface of red blood cells. Type B blood, for example, has B antigens on the surface of red blood cells. Type AB has both A and B antigens on the surface of red blood cells. Type O, I like to think of it looking like a 0. It doesn't have A or B antigens. It's naked. Well, okay, it does have other proteins on its surface, but not A or B. So think of O as looking like a 0. Doesn't have A or B. So if you have type B blood, you have B antigens on the surface of red blood cells. That means a person with type B can accept another person's type B blood because B is an antigen that their body recognizes. But if you try to give that person a type A blood type, that's an antigen that the immune system does not recognize. That person's immune system would attack. It would also attack AB blood because that includes the A that it doesn't recognize. Now type O would be safe. Remember O, think of like a 0. It doesn't have A or B antigens, so type O can donate to everyone. Now while O individuals can donate to everyone, they can only receive blood from another type O. Because type O doesn't have A or B antigens, so their immune system will attack any other blood type that does. Neither of us have type AB blood, but we would think this is a cool blood type to have in the sense that you could receive blood from anyone. If an AB person received blood from a person that had type A, well, they've got the A, so it's all good. And if an AB person received blood from a person that had type B, well, they've got the B antigen too, so it's all good. They can receive blood from type O too because there's not any antigens to even worry about. Now, one thing we do want to add that makes all of this a bit more complicated. Blood types also have a plus or minus sign listed by the blood type. This makes a big difference with blood donations. If you have a plus, it means that you have this other little protein called Rh factor on the surface of your blood cells. And if you have a negative, it means that you do not have this little protein called Rh factor on the surface of your red blood cells. Now, we're not going to be able to go into that in this short clip, so to the Google for that if you would like to learn more about this. Blood type is genetically inherited, and a great example of multiple alleles. Remember that alleles are a form of a gene, like a flavor. Just an analogy. So let's put this into practice. Let's say a couple gives birth to a baby boy. Both parents have type A blood, but then there is a mix-up at the hospital. And now, sorry, I get really into my drama. There are these two baby boys, and the hospital does not know which one belongs to the couple. Probably not a hospital where you would want to have kids, but let's try and use our blood type genetic problem-solving skills to help us out here. Baby Phil has type B blood, and baby Sylvester has type O blood. Could either of these babies belong to the couple who both have type A blood? Well, let's talk about phenotypes and genotypes. The phenotype of type A blood is A, but the genotype is written like this or this. The format of writing here helps with multiple alleles like blood type problems, and we can show you why when we work this out in the Punnett square. Now you may notice that I said that blood type A could be written this way or this way. Without testing, we don't really know which one it is. You can consider this one to be homozygous and this one to be heterozygous. The phenotype of type B blood is B, but the genotype is written as this or this. The phenotype of type AB blood is AB, and the genotype is written this way. There's no other way to write that one. The phenotype for type O blood is O, and the genotype is written like this. Remember how I said that the O kind of looks like a 0, and so you can think of it as having 0 blood type antigens? Well, that's what it is. Little I, no coefficient. Okay, back to the babies. We are told the parents both have type A, but remember, we don't know whether that means they are this type A, the homozygous, or this type A, the heterozygous. And the mom could be one of those genotypes, and the father could be the other genotype, or they could both be the same genotype. We just don't know. Okay, so let's try it out with all of the possibilities. Remember that we write the parent genotypes on the top and the side of the Punnett squares, like this. We'll fill in the Punnett squares, and we'll make sure to have capital I's first, just for formatting purposes. Okay, so is it possible to get baby Phil, who has type B, in any of these offspring? Nope, he must be someone else's baby. What about baby Sylvester with his type O? Yes, it is possible, but both parents would have to be heterozygous A. That means they both must be this. then yes, you would have a 25% chance, that's 1 in 4 here, of having a baby with type O blood. Of course, if I was advising this couple, I would also insist on a DNA test, as this blood type problem only shows that it's possible that baby Sylvester is theirs. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Linked gene. For a linked gene, each chromosome carries a number of genes in particular locus. The diagram shows the difference when the genes are linked or not linked. When the genes are linked, the majority of the offspring have the same combination of characteristics as their parents, while a few show new combinations. Please watch the next video to understand more.
That is, on the same chromosome. Here, both the color and size genes are located on the same chromosome. Again, both homologs are depicted here, and each one has undergone replication. Here, both dominant alleles are paired together on one homolog, while both recessive alleles are paired together on the other homolog. This arrangement of alleles is called a cis, or coupling arrangement. Now, because the genes are linked, the big B and big R alleles are essentially stuck together, while the little b and little r alleles are also stuck together. As a result, most of the gametes produced by the heterozygote parent are either big B, big R, or little b, little r, and these gametes are produced at relatively equal frequencies. However, during meiosis, a process called crossing over, or recombination, which occurs at relatively low frequencies, allows for the generation of recombinant offspring. You can see here that crossing over allows for the formation of the two types of recombinant gametes, one with big R little b, and the other with little r big B. Note that in this scenario, as in the scenario where the genes are located on different chromosomes, the same 4 classes of offspring are formed. However, because crossing over occurs at a relatively low frequency, the parental offspring outnumber the recombinant offspring. Here, since all offspring inherit a chromosome carrying 2 recessive alleles from the homozygous recessive parent, the chromosome inherited from the heterozygote parent determines whether the offspring are parental types or recombinant types. Parental offspring inherit a non-crossover chromosome from the heterozygote parent, while the recombinant offspring inherit a crossover chromosome from the heterozygote parent. The exact proportion of recombinant offspring formed is dependent on the frequency of crossing over between the 2 genes during meiosis, and this in turn is dependent on the distance between the genes. The closer the 2 genes are, the less likely crossing over is to occur between the genes. Now consider the alternative arrangement of the 2 genes in the same chromosome. In the trans, or repulsion arrangement, each homolog carries 1 dominant and 1 recessive allele. This arrangement is similar to the cis arrangement in that the same 4 classes of offspring are generated. However, the 2 classes of offspring that were of the parental type in the cis arrangement are now the recombinant type in the trans arrangement, and vice versa. Nonetheless, whether the alleles are in a cis or trans arrangement, we should still see to expect the parental offspring outnumber the recombinant offspring. Therefore, when faced with the task of determining the chromosomal arrangement of 2 genes, that is, whether they're linked or not linked, we can perform a test cross. If the 2 genes are on separate chromosomes, all 4 classes of offspring will be produced in equal proportion. When the two genes are linked, the two parental classes of offspring will outnumber the two recombinant classes of offspring. It is even possible, in cases where two genes are very tightly linked, that only offspring of the parental type will be produced.
Sex-linked gene. Sex-linked gene is genes that are carried by either sex chromosome, X, or Y chromosome. The example of sex-linked gene is color blindness. In this diagram, the normal vision female, color blindness carrier is married to normal vision male. There are 4 possible offspring will produce, which is normal vision female, normal vision female, but carrier for colorblind, normal vision male, and colorblind male. Another example for sex-linked gene is hemophilia. In this diagram, father with hemophilia is married to mother that carrier for hemophilia. The possible offspring will be produce is 25% son without hemophilia, 25% daughter with hemophilia, 25% son with hemophilia, and 25% daughter carrier of hemophilia. Please watch the next video to understand more.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cytoplasm that help stop us from bleeding. They help our blood clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets because special clotting factor proteins that are needed to work with the platelets may not work correctly. So even a basic cut could be dangerous for people with hemophilia because they could bleed continuously. We have many treatments for the symptoms of hemophilia, and this has greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it's different from the basic Mendelian genetic problems. What do I mean by that? Well, we still use the terms dominant and recessive for alleles, but this time, because it's sex-linked, the alleles are on sex chromosomes. This is the case with sex-linked traits. So what is a sex chromosome? Well, recall that humans have 46 chromosomes. Chromosomes are made up of DNA and protein, and they contain your genes. Well, 2 of your 46 chromosomes, they are called the sex chromosomes. In a karyotype, it is usually the last 2 chromosomes that are the sex chromosomes. The sex chromosomes are called X and Y chromosomes, but it has nothing to do with the shape of the chromosome. That's always kind of confusing, but please don't think that Y chromosomes are shaped like a Y and X chromosomes are shaped like an X. This used to always confuse me. It has nothing to do with their name. The reason they got their name is actually kind of interesting, so to the Google for that. Everyone has an X chromosome, but if you have another X chromosome, meaning you have 2 X chromosomes, you are female. And if you have a Y chromosome, meaning you have an X and a Y chromosome, you are male. There are also genetic disorders where you can have extra copies of sex chromosomes, but we will not go into that in this clip. Sex-linked traits are traits that are specifically on the sex chromosomes. Most sex-linked traits tend to be on the X chromosome because it is larger than the Y chromosome and it contains more genes in the Y chromosome. The disorder hemophilia is like this. Now we will use the letter capital H to represent an allele for having normal blood clotting function, and a letter lowercase h to represent an allele for having hemophilia. Hemophilia is a sex-linked recessive disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the sex chromosome as a superscript, like an exponent. A woman that does not have hemophilia could have the genotype X big H, or X big H, X little h. because as long as she's got at least one dominant allele, that dominating allele will be what shows. So no hemophilia, since again, hemophilia is a recessive sex-linked disorder. The only way for her to have hemophilia would be the genotype XhH, XhH, because only when there is no dominant present will that recessive show up, at least in this type of trait. Now for a male to not have hemophilia, his genotype would have to be XhY. Notice how I did not put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If he has the genotype XhY, then he has hemophilia. He doesn't have two X chromosomes, so he cannot be a carrier. So let's say that two people that do not have hemophilia have children. However, let's say that the woman is a carrier. That means she is heterozygous. How do you do a sex-linked Punnett square cross for this kind of trait? Step 1. Identify the genotypes of the parents. So the mother was a carrier. She is XhH, Xh. She does not have hemophilia because of that dominant allele present, but she is a carrier. The male, if he does not have hemophilia, well, he must be XhY. There's no other option for him. Step 2. Place one parent on the top, outside of the square like this, and place the other parent on the left, outside of the square like this. Step 3. Cross them. For formatting purposes, place X chromosomes before Y, and you also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. The phenotype ratio, remember that these are the observable traits, can be written out that there is a 75% chance that the child will be born without hemophilia and a 25% chance that a child would have hemophilia for this boy here. You could also make this a ratio, so 3:1. Notice that in this type of example of a sex-linked recessive disorder, males are more likely to inherit this disorder because there is only one X chromosome present. This is true for many other sex-linked recessive disorders, such as colorblindness. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Polygene. Polygene is an additive effect of two or more genes on a single phenotypic character. Traits that show continuous variation are not determined by a single gene, but a large number of genes at different loci. The example polygene is skin color and body height. Please watch the next video to understand more.
Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen? There isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, big C, little c, and maybe more genes to ultimately determine your height. And you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition growing up can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics.
Okay. Now we will move on the last subtopic for genetic inheritance, which is 4.3 genetic mapping. What is genetic map? Genetic map is a graphic representation of the arrangement of a gene or a DNA sequence on a chromosome. So, what is genetic mapping? Genetic mapping is process locating the relative position and sequence of gene on a chromosome. It can be carried out when the genes are linked and crossing over has taken place. The relative position of genes in a chromosome can be mapped out based upon the amount of crossing over. Crossing over value, COV, is equal total number of recombinant offspring divided by the total number of offspring multiplied by 100. Unit for COV is percent, or MAB units, or centimorgan. That all for Chapter 4 Genetic Inheritance. Don't forget to scan QR code in the next slide to enrichment your knowledge. Have fun. Thank you.